This is a Pinnacle how-to video demonstrating the antibody screening and quantitation workflow. The purpose of this video is to provide an overview of the workflow, and we'll cover just the general concepts of antibody screening and quantitation. For specific functionality contained within or referred to within this video, there are specific help files and videos that you can access. Pinnacle is a unique software in that it performs robust screening and characterization of your targeted protein sequence, as well as provides quantitation in a relative or absolute form on one or many different files. When we first open Pinnacle, this is the layout that is viewed, and the user has the option of first of all selecting a specific workflow to process the data that best fits the experimental format. In this case, we will select the antibody screening and quantitation. Once this is selected, then all of the parameters that are needed to actually perform the analysis are shown on the right-hand side. If we start at the top, you can browse to bring in your specific raw file, and this can be done by bringing in one file or many different files at one time. Then you would select the knowledge base in the form of either a FASTA file, or you can copy and paste a particular sequence. For the antibody screening and quantitation, the assumption is that there is usually one or very few, let's say up to 5 different proteins or protein sequences that are going to be considered. The second assumption for the antibody screening is that you generally have a very purified or enriched targeted protein. That is, the background contribution or matrix is generally considered to be pretty low. So you've brought in your raw files in FASTA, then you would set up your processing conditions. First of all is your digestion considerations, and you would have a primary enzyme consideration as well as a secondary. So it's fairly unique in that perspective. You also can set the maximum missed cleavage sites and allow for partial cleavage at the N and C terminus. So once you've, again, added the raw file, set the sequences that you're going to process, and enable the in silico digestion to create the peptides from the base sequences, you can further allow for screening such as SNPs, glycopeptides, and currently you have the options of no consideration for N-linked glycos, looking for both modified and unmodified N-linked glycos, or only N-linked glycos at the present time. For the example shown today, we're going to look for both unmodified and the N-linked glycos. You can, of course, do an error tolerance search, and then you can specify your static and dynamic modifications by simply opening this particular window and then selecting. Finally, you have the file reading considerations, such as the resolution settings for mass tolerance and data extraction and analysis. You have peak processing, normalization, scoring, as well as your layout view that will be explained in other videos. The example that we're going to show today is a IgG digest, tryptic digest, that was prepared in 3 different conditions: a control, high pH, and then exposed to hydrogen peroxide. And there are 2 technical replicates for the particular group. And I've already processed the 6 raw files for both qualitative and quantitation, and it took about 3 to 5 minutes to get that completed. The results of Pinnacle, once you've processed the data, would come up and show in this particular view. So you would have a results table here on the left containing all of your views in a hierarchical table arrangement, and I'll show that in just a minute. Then you have a chromatographic view, a mass spectral view, and then your grouping view. And again, as we mentioned, we've got 3 different groups shown here: control, hydrogen peroxide, and pH. And this is the order that will be displayed. And again, there's help videos to show how you can change the order as needed. Again, this is a hierarchical view, but we can certainly look at things in an overview. First of all, we've got the heavy chain and the light chain for our targeted antibody, plus we've assumed 100 unassigned peaks. So the way that the software would go in and process the data is it first utilizes the protein sequence that was provided by the user as well as the digestion conditions to match any peptides that are associated to those particular protein sequences, as well as the modified forms, and then it would go in and look for the unassigned peaks. So one of the things that we can just very quickly look at, and again we've got 6 different raw files, and we can showcase all of these in one particular view, is we can look at the overview. And this just simply gives us an annotated base peak plot for all 6 raw files. And I can just simply highlight a specific region. And now the red would represent peaks that have sequences assigned to the heavy chain. Blue represents the light chain. And anything in purple, as shown here, would be for the unassigned peaks. And of course, we can hover over the main peak, and it would of course give us the peptide sequence as well as which protein it's attributed to. The other thing that we can then look at is for the specific proteins itself. If we click on the protein, we now have all possible peptides that are associated to that particular protein as well as in the legend. Depending on where we highlight a particular region, would refine the peptides to only those that are eluting in that time window. If we want to look at the specific peptides, one thing we can do is simply expand down, and there is a help video to go through the scoring. But basically, those peptides that have a red question mark simply have good MS, but may not have the strongest MS2. The blue stars represents both excellent MS, precursor isotopic correlation to the experimental, between the experimental and the theoretical, but are not showing significant changes between our different groups. And then, of course, the green trophies show good MS and MS2 matching, good integration or low variance, as well as showing some change amongst the different groups. So just quickly, if we highlight a specific peptide shown here, and of course it has a methionine, so we would expect it to show a stronger oxidation from the hydrogen peroxide samples, we can of course simply look at what the results are. So of course, one of the overviews in Pinnacle for the antibody screening and quantitation is that I can simply look at the table and get a view or an overview of what's happening for each individual peptide through these gel views. So I have a file ratio view, which simply shows me sort of a gel view plot for the relative abundance across all 6 files. And they're arranged based on our grouping control showed here. So the first 2 files would be attributed to the control, the next 2 would be to the hydrogen peroxide treated samples, and the last 2 for the hydrogen peroxide or the higher pH. And then of course the average values would be contained in the group ratio view. So we can again look to see if all of our individual files are behaving in a systematic manner, since these are technical replicates for the different groups, as well as what's the average view. And if we just look at these 2 peptides, which is the showing the oxidized and the unoxidized form, we can of course see that, as predicted, the oxidation in the hydrogen peroxide treated samples show a greater abundance. Of course, we can certainly zoom in and just hover and get precursor information at any point in our eluted or our XICs. And the triangles again represent a matched MS2. So if we just simply click on one, we get, of course, the product ions for a spectrum assigned to this particular peptide. And we can show a multiplot for the oxidized form then. And again, zoom in and just simply show the comparison and evaluate if in fact our mass differences can be localized to the specific residue. So in addition to evaluating those peptides that are attributed to the protein sequences and looking at the gel views, Pinnacle also does this for our unassigned peaks. And if we expand out on the unassigned peaks, again we have those unassigned peaks that are behaving in a consistent fashion across each raw file versus those that show some form of differential expression. And again, depending on the known peptides and how they're behaving across the different groups, we can use this information to start trying to identify if these particular unassigned peaks could be attributed to one of our proteins or another. And of course, The other aspect in total characterization is not only are you interested in which peptides, and of course the sequence coverage for the heavy and the light chains, but we also have ways to change the different views. And one particular view that is extremely beneficial for sort of antibody characterization is this protein PTM view. And in the protein PTM view, you're moving away from looking at a specific peptide sequence that's attributed to a given protein, and you're more interested in simply the site of modification. And so again, you have the residue, the location, and what's happening. So in some cases, you have deamidation as well as unmodified, or the native forms. So if we just simply click on a specific peptide, and we'll just, the methionine at residue 389 has oxidized and an unoxidized form. Once we click on just the site, we get the chromatographic views of what's going on for the all peptides that cover this particular site, and you can begin to get an appreciation for the relative abundance of those particular sites. And again, all of this is contained within the antibody and screening workflows. This, of course, ends this particular how-to video.